Welcome to Datta Biology classes. So we are discussing about photosynthesis. The other day we have discussed the role of H2O, CO2 and sunlight. These three we have discussed. Now we discuss the chloroplast. Chloroplast. So after chloroplast, we will directly enter into the photosynthesis proper. Let us start with the chloroplast right now. Chloroplast is a double-walled plant cell organelle. It is having double wall like mitochondria. So this double wall is the outer, this is the outer membrane or this is the inner membrane. These two encircles the chloroplast proper, and this is known as periplastidial space. Periplastidial space. This space is known as periplastidial space. It is divided into two parts. Mainly, these are called granum. G-R-A-N-U-M. Granum. Individual, these are granum. Are 1, 2, 3 collectively called grana. G-R-A-N-A. Grana. So this is grana, and this portion full of fluid known as stroma. Here the Kelvin cycle goes on. Okay? And in the grana, so long we have been knowing that in the grana the light phase goes on. No, it is not in the grana, it is somewhere else. Let us come. Prior to that, we have to say one thing, that these portion, these are all like a coins, pile of coins, one after another. It is there. This tail is known as granum, and this we have taken it out. Say this will be like this. This is known as thylakoid. क्या बोला? Thyla. This is known as thylakoid. So within the thylakoid, the reactions goes on. In 1905, Blackman discovered or invented that this photosynthesis goes on in two phases, and he named these two phases as light phase, and another one is dark phase. This light phase occurs in grana, not grana. Light phase occurs in thylakoid. Okay, and the dark phase occurs in stroma. But subsequently, this is a misnomer which is not given the proper meaning, because somebody may interpret that it occurs in dark, but it does not occur in dark. It takes place within the same time, that is, but here light is not necessary. So these are the two phases by which photosynthesis takes place. Okay. Now, we have to enter into chloroplast, details of chloroplast. This you have already discussed. We have already discussed 6CO2 plus 12H2O, and the reaction goes on. Light and chlorophyll, and the reaction goes is C6O6 plus, I will write 6O2, and this H2 comes here H12. 12 is to, that is 24, out of 24, 12 is here. Another 12 should be somewhere. Let's see. So C6H12O6, this is CO2 is reduced. This CO2 is here, C and O. CO2 is here and hydrogen is donated here. It is hydrogen donor and hydrogen makes the glucose. Then 6O2, it has been proved, last day we have discussed, it has been proved by O18 isotope that the entire oxygen comes from water. Entire oxygen comes from water. The here are 12 H2O, 12 H2O. Out of 6 to 12 H2O, that is 12 O done. Out of 24, 12 to 24, we have received 12 here, another 12 left. So that 12, what we do? That 12, 6 H2. That is that 12 makes that 6 H2. And what about O? This O is 6 to 12 O. Out of 12 O, here is 6 O minus 6, then 6. 6 O. Question may be asked to puzzle you, what is the source of oxygen evolved during photosynthesis? You will definitely die. It is the source of oxygen is water. While in case of this, if they ask what is the source of oxygen in the water evolved during photosynthesis, you should say that is the source of water, oxygen in water is CO2. Okay, this was left. So this keep in mind. Now we will discuss the chloroplast. So chloroplast I will come across, that is grana, this is stroma. In the stroma Kelvin cycle goes on, and in the grana are made up of piles of points. A single point I am taking it out, it is known as thylakoid. Okay? Now this thylakoid I am writing. This is the thylakoid membrane. I am taking one piece here. This one piece I am taking out. I am writing here. It's okay? Now situated in this thylakoid membrane, I will have to make it a little bigger. Otherwise, I cannot put PS1, PS2 here. Say it is bigger. This thylakoid I have taken, it is bigger. Here it is the point, part of the point. Situated in the thylakoid membrane, a cluster of pigment molecules are there. Cluster of pigment molecules are there. The molecules are say like this. Along with chlorophyll A and chlorophyll B, the other pigment molecules are there. So let me draw the other pigment molecules. These are all different pigment molecules. Here also the other pigment molecules are there. with the other pigment molecules, there is a special pigment molecule known as P700, and here there is a special molecule called P600. This is known as, we call it as PS2. This we call it as PS1. Means photosystem 2, photosystem 1. So there are two photosystems lies within this point, that is within this thylakoid membrane. Within this thylakoid membrane, these two are available. These two photosystems are available. Now this contains somewhat 300 molecules, there it contains some 100 molecules. So all the molecules, sun ray, this is sun, this sun ray falls on this cluster of molecules. This cluster is known as PS2, this cluster is known as PS1. So all the, it will absorb light. It will absorb light. It will absorb light. And 680 nanometer or below that, 680 nanometer or below that, they will absorb the light energy or photon. So these photons are coming, it transfer it here, it transfer it here, it transfer it here, it transfer it there. They are accessory pigments. These accessory pigments molecules, and they are called antenna molecules. So there are several antenna. These antenna are receiving the photon. So whenever they are receiving the photon and or below 680 nanometer, these constitute the PS2. And what happens? They passes on this photon to main center, that is reaction center. This P680 is known as reaction center. They passes on this to 680 reaction center, and 680 and this pigment 680 ejects an electron. That electron is excited. This is the PS2. And PS700 is consists of some 100 molecules. It also receives light energy, that is photon. They receive light energy at 700 nanometer or below that. It remains, say it absorbs at 690, it absorbs at 680, it absorbs at 500. So at or below the nanometer, they are receiving, they are antenna molecules. They passes on that energy, I mean photon, to the main reaction center or photocenter or P700. So P700 is the main reaction center. It ejects electron. Its electron is excited. These are the photosystem 1 and photosystem 2. These are the reaction center or light harvesting center. Or one thing we like to mention, that whenever they are ejecting one electron, so that energy, how much energy is required to eject an electron from 680 or 700, that quantum of energy is known as quantojohns, and that is why the name is quantojohn comes. So these are the two photosystems available in the thylakoid membrane of the grana. These are known as FRET, F-R-E-T, FRET, or intragranular lamellae or stroma lamellae. Stroma lamellae. Stroma lamellae. So the light phase or the light dependent phase, that is, this reaction depends on the light, because until and unless light falls here, it cannot eject electron and the reactions cannot goes on. So this is light dependent reaction. It occurs in the thylakoid membrane of the grana and it occurs in the sunlight. Now I am taking this in a bit explanatory way. Give me just 1 second. This out of this grana, I have taken one thylakoid here. Side of the coin. I am taking the side of the coin like this. I have taken this, writing in this way. This is the side of the thylakoid membrane. In the thylakoid membrane, there are embedded some proteinaceous structures. Say this is a structure. This is known as PS2. This is known as PS1. Okay? This PS1 consists of what I told you, that 680 pigment is there, along with other photosynthetic pigment or antenna molecule or light harvesting molecules here. And here it is 700, P700, I am writing simply 700. And this antenna molecules or light harvesting molecule or accessory pigments or associated pigments are here. Inequality in any way. So what happens? Whenever light falls here, sunlight falls here, the reaction takes place. I have taken that one and keeping it in this way. It is embedded in the thylakoid membrane. And outside this, there is the stroma. There is stroma. Okay? Now, whenever light falls here, here starts the reaction. Main reaction, it starts here. Whenever the light falls here, that is photon hits here, one electron is ejected, excited. Because the electron gets excited with that quantum of energy. This electron gets excited. Okay? First phase. So, this phase is known as, where is sun? Number 1 is known as, I am writing here, Photoexcitation. So whenever light falls on the cluster, it ejects one electron. It is known as photoexcitation. Here from the sunlight comes. Sunlight is coming. Number 1. This photoexcitation helps to eject one electron from the 6 empty molecule. That electron gets excited. It cannot stay in this place. So it passes on to primary acceptor or electron transport chain. This electron passes on through a series of electron transport chain. This is known as ETC. Electron transport chain, ETC. So this electron passes on through a electron transport chain. This I am marking at, this is 1, I am marking this one as 2. It is known as electron transport chain. So what you will write? Photoexcitation. Then electron passes on through electron transport chain. and finally the electron reaches here. It doesn't go anywhere. Why? Because electron while transporting from one electron carrier, it first accepted by the primary acceptor, then this electron carrier, this electron carrier, this electron carrier. So in name, I will take a different episode or different video on the PS1 and PS2. But here you take it in mind that this electron passes on this electron carrier and reaches here, because while passing through the electron carriers, it loses almost all of its energy. So it stays there for a while. And while passing, the energy, some energy is liberated. Some energy is liberated here. So that energy helps to remove some proton inside the thylakoid. Inside the thylakoid, increasing the proton gradient within the thylakoid membrane, because it is thylakoid membrane outside the stroma. So within the thylakoid membrane, the plant is increasing its proton gradient. At the same time, what happens? This is first, this is second. At the same time, what happens? Here I am writing. H2O is splitted. H2O is splitted, 2H, H plus O. In these three components, it is splitting. Whenever H plus is splitting, it contains one electron also. This is proton, and outside one electron also. And this O, that is half O2, we can say it is O, that is half O plus H produces H2O, or this O actually goes out in the form of oxygen. This is the oxygen liberated through the stomata. This is the source of photolysis of water. This oxygen is pulled out through the stomata. And what happens? This is known as, this reaction you will write first, second, third. Third one is, you write photolysis of water. Photolysis of water. That is, with the help of photolysis, breakdown takes place of water. Water is broken down into H plus or H minus ion. Here this oxygen goes out finally. And what happens? Once this electron is ejected from 680, it gets excited because it creates an electron hole into its molecule. So that has to come into ground state or stable form. So this electron comes here to replace the electron lost from here. This replaces the electron. This is photolysis of water. And what happens next? Next what happens? This is the proton gradient increasing inside the thylakoid. It is highly acidic. So another protein bound complex is there. This complex is known as ATP synthase. SYNTHAC. ATP synthase. These are pushed on because these hydrogen and H plus ions are increasing. So this is pushed through this area. While hydrogen and proton gradient are passing here, that is capable enough to fix inorganic phosphate with ADP, adenosine diphosphate. Taking this inorganic phosphate is converted to ATP. That is the synthesis of ATP. In the life phase, we are producing two things which is required for next phase. This is the main products of this light reaction. This process that is pushing the hydrogen ion through the ATP synthase. You will write, you write 1, 2, 3, 4. 4 you write 1, 2, photolysis 3, and this is 4. You write chemiosmosis. Through a process known as chemiosmosis, the proton gradient is decreased, proton gradient is passes onto the stroma, and while these protons are passed to the stroma, it releases energy. That energy is capable enough to produce ATP by adding one inorganic phosphate. So ATP has been produced. Next, this ATP is produced, you write it 1, 2, 3, 4, 5. Phosphorylation, or reduction. Phosphorylation or reduction. 5. These are the PS1, PS2, sorry. These are PS2. And whenever light is falling on the PS2, light is also falling here itself. So here light, that is photon, is falling over here itself. So these the antenna molecules or light harvesting molecules are harvesting or trapping the photon here. They are absorbing at 700 or below 700. They are absorbing the light energy. That is photon they are absorbing. They are transferring also, they are the same way they are transferring. And this 700 also get excited, it rejects electron. So I am exciting this electron also. This electron I am exciting. This electron, PS1. Inorganic phosphate, ATP. These electrons then also pass through electron transport chain. These electron transport chains are like this. Here is one electron transport chain. Here is another. Here is another. There are 3 electron transport chains here. I am writing. This electron is jumping from electron transport 1, electron transport 2, electron transport 3, ultimately passes out through the thylakoid membrane to the stroma. And this electron transport chain is known as ETC. This is 5, this is 6. 6 ETC. So what you will write? Another case of photoexcitation takes place, and this photon hits the cluster of molecules of PS1 containing the P700 molecule and along with the other accessory pigments, and which helps to eject one electron, and that electron is excited. That electron again passes on to the electron transport chain, and ultimately the electron passes on to stroma. And this electron reduces the NADP plus to NADPH plus H plus. So here another important product required for the second part of the reaction. These are the 2 products, main products, for which we are looking for, because we are producing, mainly producing C6H12O6. That is our main aim. That is, plant produces food. To produce the food, these are the system This is the light dependent phase, that is, it depends on the light. Number one is light dependent phase, LDP, not long day plant, light dependent phase, number one. And number two is light independent phase. Light independent phase does not require light, but it occurs in the same time, in the same place, in the stroma. So this is the part one. And in the stroma what happens, that will come in the next episode. Thank you very much. Till then, goodbye.